Translation in eukaryotes has a lot of molecular mechanistic similar to that in the prokaryotes, and the only difference being that there is a certain degree of complexity associated with eukaryotes, which is taken care of by several different initiation factors. So, this session is going to focus mainly on the initiation phase of translation in eukaryotes. So, let us look at the learning outcomes of this session. The phase of initiation comprises assembly of components like the small subunit of the ribosome, tRNA, mRNA, along with initiation factors to form what is called as the pre-initiation complex, which gradually, of course, moves to forming the initiation complex with this larger subunit, which is the 60S in eukaryotes. The sequential and coordinated complex formation is due to the various initiation factors, and the initiation phase is also an important phase of regulation of translation. The initiation complex formation commences at the 5' cap of the mRNA, and thereafter there is scanning of the mRNA to identify the start codon. Understanding of the molecular mechanistic of the process has helped identify several biological targets for therapy or diagnostics. Let us first begin with what we understand by pre-initiation complex, which is also termed as the 43S complex. Interestingly, the pre-initiation complex from 43S moves to 48S, and after which only you have the initiation complex being formed. Now, the beginning or the formation of the 43S complex begins with the formation of what is called as a ternary complex, and the ternary complex is basically coming together of three components, which is one, the eukaryotic initiation factor 2, GTP, and the initiator tRNA, which of course is charged with the methionine. Interestingly, the initiator tRNA structurally has very distinct characteristics that are different from all the other tRNA, and also especially different from the elongated tRNA for methionine. So, when you look at the elongated tRNA for methionine and the initiated tRNA for methionine, there are three very obvious differences that distinguish a initiator tRNA from an elongated tRNA. The first one is, of course, the presence of a AU base pair in the acceptor arm of the initiator tRNA. And in fact, several studies have shown that if one replaces the AU with a GU, okay, these studies have been carried out in yeast, and when you have a GU in place of AU, it was found that the binding of the initiator tRNA to the EIF2 decreased tenfold. And so, it would not form the ternary complex at all. Another distinctive feature is that in the T loop, or rather the T loop of the human tRNA initiator is very different from the T loop of the elongated tRNA for methionine. For example, here you have the residue A60 at the 60th position being replaced by U. So that is one thing which is very different. Then you have the 54A being replaced by T. And also you have a pseudo-uridine over here, whereas here you have the uridine itself. So these are certain differences that are observed in the T loop of the initiator tRNA. Now, interestingly, what has been also observed is that the initiator tRNA at the anticodon arm has three consecutive GC base pairs. And this, if replaced or if modified, then it has been shown to have decreased activity as the initiator tRNA. And these studies have been done in which in the elongated tRNA, when the AU is replaced by GC, you would have 3 consecutive GC formed even in the elongated tRNA, and thereby it has been observed that this elongated tRNA could function as the initiator tRNA. So these are certain interesting aspects of the structure and the functions of the initiator tRNA and the elongator tRNA. Now let us go to the second component, important component of the ternary complex, which is EIF2. EIF2 is basically made of 3 subunits, alpha, beta and gamma, and basically the gamma subunit is able to bind to GTP. So you can see that the GTP is interacting with the EIF2 through the gamma subunit, and this gamma itself is able to bind to the initiator tRNA. Moreover, the beta subunit also is able to directly bind to or interact with the initiator tRNA. So that is how you have the active ternary complex of EIF2, GTP, met tRNA, initiator being formed. Please note that the presence of GTP is important for it to make it active. Therefore, if you have an EIF2 complex with GDP, then the EIF2 GDP does not have a good affinity for binding to the initiator tRNA, and therefore a ternary complex will not be formed. Now, here is where another molecule comes into prominence, and that is eukaryotic initiation factor 2B. This molecule is a nucleotide exchange factor. So, for EIF2B, EIF2 is the substrate, okay, and its catalytic activity enables it to exchange the GDP if bound to EIF2 with GTP. So it can convert an EIF2 GDP to EIF2 GTP. And so if EIF2B is present, then you would have a formation of more of EIF2 GTP, which in turn will be able to bind to the initiator tRNA to form the activated ternary complex. And hence EIF2B is a point of regulation. So it has been observed that when certain kinases phosphorylate EIF2B, the phosphorylated EIF2B is not able to interact with the EIF2 GDP and convert it to EIF2 GTP. Therefore, a phosphorylated EIF2B is not able to form or allow the formation of a ternary complex, and hence no formation of 43S complex, and hence no formation or no initiation phase of translation. It has also been observed that if the EIF2 is phosphorylated, as you can see over here, the alpha subunit of the EIF2 if phosphorylated, EIF2B is able to bind to the EIF2 GDP, but it is not able to convert the EIF2 GDP into EIF2 GTP. So phosphorylation of either EIF2B or that of EIF2 itself is making it inactive complexes, and therefore you will not have ternary complexes being formed. So phosphorylation is actually regulating the formation of the ternary complex. And so you can see that at the very first step of the translation initiation, you have already regulation. To look into it a little, with stress, what has been observed is that the phosphorylation of EIF2 increases. To look at it a little further, EIF2B exists as a decamer. So basically, you have EIF2B present in such a way that there are two units, or a doublet of every uh, subunit present, which means that you will have 2 alpha. So you can see that this red over here is alpha. So you'll have 2 alpha. You will have 2 beta. The blue is 2 beta. You will have 2 gamma. So the green is the gamma. You'll have 2 theta. So the golden subunits are the delta, and you have 2 epsilons, which is the gray ones. So therefore 2 of gamma, 2 of beta, 2 alpha, 2 delta and 2 epsilon forms a decamer structure. Now it is interesting to note that if EIF2B is a decamer, then it has an enhanced NEF activity. NEF means nucleotide exchange factor. Now, of all these subunits, it is the epsilon which has the catalytic activity, and of course EIF2 is the substrate. Now, considering the fact that EIF2 on phosphorylation, specifically at a serine 51, which is conserved, the EIF2 itself becomes a competitive inhibitor for the catalytic activity of the EIF2B. So basically we have understood that if EIF2 is phosphorylated at its alpha subunit, specifically at serine 51, then EIF2, although it binds to EIF2B, does not allow its catalytic activity. So, in short, therefore, what is going to happen because of EIF2 alpha becoming phosphorylated, protein synthesis will stop. So, when does this condition arise? So, it has been observed stresses like misfolded proteins, viral infections, cells redox state, etc., are able to uh, activate certain kinases, and those kinases in turn are able to phosphorylate the serine 51 of the EIF2 alpha. So therefore, you can very well see how you have the protein synthesis getting regulated by conditions such as stress conditions as well through phosphorylation. Now let us go to look at the next initiation factor which helps in the formation of the 43S complex. So far what we have understood is that a ternary complex is formed when you have EIF2, GTP and the initiator tRNA coming together. Now this ternary complex has to bind to the small subunit which is the 40S, and it has been found that EIF3 is able to basically bring the ternary complex close or attaching it to the 40S subunit. Now, EIF3 in itself is the largest and the most complex molecular complex in the pre-initiation complex. So it has been observed that EIF3 itself has about 11 to 13 non-identical subunits, and because it has these many different subunits, it would have multiple functions. What are the functions that are involved? And let us look at how you have the 11 to 13 non-identical subunits. So EIF3A, B, C, D to M is what the different subunits are. And what has been observed is that out of the 13 non-identical subunits, 8 of them form a very characteristic shape, as you can see over here. And this is very similar to certain other family of molecules. But what is very clear is that the EIF3 is able to bind to the platform of the 40S. And it is also a scaffold to bind to different other than EIF2 or the ternary complex. It is also able to bind to other initiation factors. So of the many functions that the EIF3 shows, one of the main functions is that it controls the binding of the ternary complex to the 40S. It positions or helps position the mRNA on the 40S. So we all know that for initiation of translation, the binding of the mRNA to the ribosome is very important. So you can see how EIF3 has a very important role in positioning the mRNA to the 40S subunit. It also modulates the start codon selection. So just having bound to mRNA is not enough. What the pre-initiation complex also has to do is to identify the start codon and then position the met tRNA, that is initiator tRNA, on the start codon. Now, the EIF3, as mentioned, also acts as a scaffold to bind to various other initiation factors. And interestingly, what has been found is that when EIF3 is bound to the 43S complex, then it doesn't allow the 60S to disassemble the 43S complex. So in a way, EIF3 is stabilizing the 43S complex and also keeping the 60S away from the 40S itself. If you remember, in the prokaryotic system, this job was being done by IF3. So IF3 was responsible to binding to the P side, to the E side, and not allowing the association of the 30S and the 50S. So here what is understood as the fact that EIF3 is also doing the same thing. Now let us understand that the 43S complex, as it is forming, it is going to initiate the translation, but in phases, in steps, and this is actually dependent on the 5 prime cap that of the mRNA. So when initiation takes place with the help of the 5 prime cap of the mRNA, then it is called as cap dependent initiation. Now, unlike in the prokaryotic system where the 16S rRNA is able to recognize the Shine-Delgado sequence in the 5' untranslated region of the mRNA, in case of the eukaryotic system, the 18S rRNA does not have any complementary sequence that can bind to a Shine-Delgado sequence. And neither is there a very distinct sequence such as Shine-Delgado sequence in the eukaryotic mRNA. In fact, the eukaryotic mRNA would have something that's called as Kovacs sequence, but other than that, it does not have Shine-Delgado sequence, and hence the binding or the positioning of the AUG on the on the P side is not because of the mRNA per se or the ribosome assembling or at the or the 16S rRNA helping the mRNA to lodge. It is more dependent on the 5 prime cap of the mRNA. So one of the ways of recruitment of mRNA to the 43S is using the 5 prime cap. And this is aided by EIF4 factors. And there are several different EIF4 factors: EIF4A, EIF4B, E, G, H. Now, EIF4E is an essential EIF4 factor for translation initiation, and it is also conserved across species. So you will find the same in yeast, you'll find the same in humans, you'll find the same in protozoans, etc. Now, what it does definitely is that the EIF4E interacts with the 5 prime cap. All of you know what the 5 prime cap is. So the 5-methyl guanine that is added to the 5 prime end of a mRNA is what the 5 prime cap is. And this 5 prime cap not only enables, as you can see, initiation of translation, but it also enables transport of the mRNA from the nucleus through the nuclear pore complex into the cytosol, and it also has found to be having a role in stabilizing the mRNA per se. Now, the interaction that EIF4E makes with the 5 prime cap is aided by the EIF4G. This EIF4G is a multifaceted protein in the sense that it has several domains that enable it to carry out several functions at the same time. So it has domains that can interact with EIF3. We already know that EIF3 is the one that brings the ternary complex to the small subunit. It is able to interact with the EIF4A, and it is able to interact with the mRNA also. Moreover, what has been observed is that its N-terminal domain can bind to poly-A binding protein. We all know that mRNA in eukaryotes have a 3' poly-A tail. Those poly-A sequences that are present at the 3' end of the mRNA are always bound to by the poly-A binding protein. And those proteins can in turn bind to EIF4G. So, in a way, what we can understand from this is that the EIF4 factors of the 43S pre-initiation complex are able to bind to the 5' end of the mRNA as well as to the 3' end of the mRNA. So it is interesting to note that both ends of the mRNA are brought close to each other thanks to the EIF4 factors. EIF4G also has a few isoforms, that is, you can have EIF4G1, EIF4G2 that way. EI4A is a RNA helicase and it belongs to the family which has the DEAD conserved sequence in its structure. Now, here EI4A has three subforms A1, A2, A3. And of these three, A1 and A2 play a role in translation initiation. So it basically ensures that it has a weak helicase activity. So it will be using ATP as well. But the helicase activity is weak. Now, what is the role of the RNA helicase? So if the mRNA that is bound to the 40S has secondary structures, then those secondary structures are resolved by the helicase. So it is therefore an RNA helicase. Now, what is interesting to note is that the helicase activity is enhanced when you have a complex called EIF4F formed. And this EIF4F complex is formed when EIF4A, EIF4E, and EIF4G form or come together. So the EIF4F, that means a combination of 4A, 4E, and 4G, makes the helicase activity greater than if you have only EIF4A. EIF4A3 does not seem to have an important or essential role in the translation initiation. EIF4B is able to stimulate the EIF4A, and it is also observed to be able to bind to RNA. So the idea is that probably EIF4B binds to not just the 18S rRNA, but it also binds to the mRNA. So this is another important aspect. Now EIF4H per se is able to stimulate EIF4A. Other than it stimulating the weak RNA helicase activity, there are many studies that suggest that EIF4H is a protein that is used by the viruses to come in to have their RNA their RNA translated. So the virus machinery uses the machinery of the host system to transcribe and translate their genetic material. And so EI4H has been associated with several viruses thereby. Moving further, just to understand how EI4E and 4G as a complex work well, so you can see that the EI4E has a globular region with a flexible tail coming out of it. Now, you will find that when EI4G binds, there is a slight folding of the tail of the EI4E, and with this folding of the EI4E tail, you will see that there is a slight change in the conformation of the EI4E, exposing that part of the EI4E that can bind to the mRNA. So this is the part of the EI4E that can bind to the 5' cap of the mRNA. So as you can see, where here it is more hidden, here it is more exposed. And so binding of the EIF4E to the mRNA is enhanced by binding of the EIF4G to the EIF4E. So therefore you have now an interaction over here, and also interestingly, the binding of the EIF4G to the EIF4E also ensures that this mRNA is also bound to the EIF4G. So the tail is bound to the EIF4G, whereas the head or the 5' cap is bound to the EIF4E. This is possible because the EIF4G has a domain that can bind to the poly A binding protein. So you will find these actually associated with poly A binding protein, and therefore they can very easily bind to the 3' end of the mRNA as well. Now, interesting to note is this, that the EI4E actually is present within the nucleus in an inactive form. And it is kept inactive because it binds to 4E binding proteins. So the binding of the 4E binding proteins keeps the 4E inactive. Now, only when the 4E binding protein is released and in place of that you have EI4G coming and binding, you will have the EI4E becoming active. So you can see here, you cannot see the region that binds to the 5' cap of the mRNA, but look at this, you can immediately find the region that binds to the 5' cap exposed. And so this can bind effectively to the 5' cap, whereas this cannot. Now the question here is, how does the EIF4E become active? So what has been understood is this, that phosphorylation is again a mode of regulation, and so when EIF4E is needed to be active, then it becomes, or there is a phosphorylation by MNK1, which is actually also bound to the EIF4G, and that helps phosphorylating specific amino acids on the 4E, releasing the 4E binding protein, and in place of the 4E binding protein, at that position, you will have the EI4G coming and binding. So there is a conserved serine that is present, that is phosphorylated by MNK1, and thereby you have this EI4E, EI4G complex form. The next step is scanning. So now what we have is that the 43S complex is bound to the 5' cap of the mRNA and also the 3' poly A by 3' tail of the mRNA. Now what the 43S complex has to do is it has to scan the mRNA at the 5' UTR region. So where it is bound at the 5' cap, from there, that point on, it has to basically move and try to locate the start codon. So if this is the 5' cap and this is the 43S, the 43S will have to scan this region. This region is what is called as the 5' untranslated region, and it'll have to scan until it reaches the start codon, until it identifies the start codon. So invariably, therefore, the initiation complex that scans the 5' UTR until the initiator tRNA base pairs with the start codon AUG, such a model is what is called a scanning model. And there are several studies that point to the existence of this scanning model. For example, it has been observed that translation efficiency decreases if the 5' UTR has a stable secondary structure. So you can see if here there is a secondary structure, then the 43S pre-initiation complex is not able to move beyond the secondary structure. So this acts as a barrier. In that case, it is never going to reach the AUG and start translation. So therefore, you can understand that it is scanning, and because it is not allowed to scan and locate the start codon, you don't have translation being efficient. The second thing that has been done is that the mRNA that already has a start codon, to that mRNA, if another start codon is introduced before, that means placed upstream of the actual AUG, then it has been observed that the translation start site that is marked is different from the actual translation start site. So you may understand that the ORF changes, and so therefore what is understood is that if this is the AUG and you have an AUG added in the 5' UTR region, that AUG is being identified because the 43S complex is scanning. So this is what is understood. You have the 43S with the met tRNA coming. When it reaches here, the tRNA will base pair with the start codon, and from that point on, translation initiation will begin. So where the ORF should have begun here, now the ORF is beginning here. So this is basically trying to say that scanning and locating the start codon is carried out by the 43S complex. But of course, 43S complex is not the complete pre-initiation complex. The 43S will in fact bind to several other EIFs, leading to formation of the 48S complex at the start site. So let us look at what forms the 48S complex, or which are the eukaryotic initiation factors that will form the 48S complex. So EIF1 is one component that promotes the correct positioning of the 43S on the start codon. You have another component called the EIF1A. This too enhances the positioning of the 43S onto the start codon. And EIF1A also interacts with EIF5B. And so what is observed is EIF5 and EIF1 can also possibly indirectly or not directly associate with the ternary complex. That means with the EIF2 GTP initiator tRNA. So EIF3 binds both EIF1 and EIF5. And interestingly, the EIF1A occupies the A site of the 40S. Since it occupies the A site of the 40S, no cognate tRNA can come and bind to the pre-initiation complex. So there is only one tRNA that can now bind to the ribosome, and that is the initiator tRNA, and that too has to be positioned at the P site and not the A site. And so that is taken care of by the fact that the A site is blocked by the EIF1A. EIF5B is also directing the initiator tRNA to the P site. So together EIF1A and EIF5B ensures that the initiator tRNA is positioned at the P site. So this is how it is. You can see that this is the ternary complex with the anticodon binding to the AUG. The 5' cap of this mRNA is bound to IF4E. You will find the IF4A resolving secondary structures of the RNA, and you can see how the IF4G is able to bind to EIF3, it is able to bind to RNA, and it is also able to bind to the poly A binding proteins. So you can see the 5' cap is close to the 3' end of the mRNA. So you have the entire mRNA circularizing to have the 3' poly A tail very close to the 5' cap. So this overall is what is understood to be the 48S complex. So you have EIF1, EIF5 and EIF1A, and as you can see 1A is covering the A site. Now when the met tRNA base pairs to the start codon at the P site, there are very distinct conformational changes in the 40S subunit. And those conformational changes are considered to be 48S post-scanning complex. And interestingly, EIF2 GTPase. So we all know that EIF2 that is bound to GTP has an inherent GTPase activity. So because of the conformational change in the small subunit of the ribosome, you have the EIF2 GTPase becoming active, and so EIF2 GTP will become EIF2 GDP, and the moment EIF2 GDP is formed, this has less affinity for the initiator tRNA, and along with 1A, it leaves the entire complex. So this is what is understood, is that the 48S, which is bound to the initiator tRNA, you can see that it is positioned in the P. You have the GTP present over here, and you have all the other initiation factors. Suppose there is a conformational change in the smaller subunit, we can see effectively that EIF2 GDP with the EIF1A is released. The moment EIF1A and EIF2 GDP are released, you will find that another initiation factor 5B would come and bind to again the platform in such a way that it interacts with the initiator tRNA, and it also brings in 60S. So therefore, this that is formed is what is called as the ATS initiation complex. So 60 plus 40, or the 60 and the 40S come together to form the ATS. Now, the moment you have the 60S carefully binding to the 40S and the EIF5B forming or interacting with the initiator tRNA, the GTPase activity of EIFB is activated and it will hydrolyze GTP into GDP. Now, in the GDP form, the EIFB has less affinity to remain bound to this 40S, and it leaves, leaving what is called as the ATS, which is ready to now initiate elongation. So this therefore is now going to be called as ATS elongation complex. And with the EIF5B getting released, what also is observed is that the A site is now free, completely free. Nothing is bound to that A site, and so you can have a cognate tRNA coming and binding to the A site. So therefore, it is ready for elongation. As the cryo-electron microscopy map shows, you very clearly can see how the fourth domain of the EIF2B, EIF5B is able to bind to the acceptor arm of the initiator tRNA. And therefore, when this interaction happens, its GTPase activity is enhanced. It is hydrolyzing GDP into GTP into GDP, and with the formation of GDP, this leaves the entire complex and goes away, leading to formation of the ATS elongation complex. Now, apart from cap dependent initiation processes, you also have cap independent initiation, but of course, this is not as prevalent as the cap dependent initiation, and it is found to be observed only in certain situations. So, it is reported to occur in stress or abnormal conditions, and one concept is the use of what is called as internal ribosome entry site. And this IRES is nothing but secondary structure present in the RNA itself, and that secondary structure is identified by the EIF4G, the ITAFs, EIF3 and 40S. So what you have over here is the formation of a pre-initiation complex, but this pre-initiation complex is devoid of the EIF4E. Instead of the role of EI4E, IRES does the job. So therefore, you don't have this 43S complex actually binding to the 5' cap at all. It simply comes and binds to the IRES region, which is very close to the start codon. And from the start codon, the translation can be initiated. Another cap independent initiation that has been reported is through what is called as cap independent translation elements or methylated 6 adenine. So you will find that at the 5' UTR region or within the mRNA there are several regions where you can have a CITE or you can have a methylated 6-methyl adenine. Now at the 5' UTR, if there is a CITE or there is a methylated adenine, then you would have the pre-initiation complex being formed there. And with the pre-initiation complex being formed there, the 5' end also comes close to the start codon. You can see how it does so. And from this point on, the pre-initiation complex scans the 5' UTR until it reaches the AUG. Once it reaches the AUG, it will form the initiation complex. So here you can see that this RNA does not have a 5' cap. It has a 5' end, but it does not have a 5' cap. And in such a scenario, you will find that the pre-initiation complex forms on CITE or the 6-methyl adenine. So this is another important feature which has been associated with many cancer cells or with also viruses that have infected a cell. So to make the conclusions, the formation of 43S complex is a regulated process that helps in assembly of the translation machinery on the mRNA. The formation of the 48S complex from the 43S enables positioning of the initiator tRNA at the marked translation start site, defining the open reading frame. The binding of 60S and stabilizing the initiation complex is aided by regulation through GTPase activity of EIF5P. The initiation complex circularizes the RNA as the former can bind to both the 5' cap and the poly-A tail, establishing also what is understood by ribosome cycling. Thus, the initiation phase of translation in eukaryotes is 5' cap dependent and orchestrated by several initiation factors that not just marks the ORF but also regulates whether protein synthesis is to begin or not. Thank you.